go ahead and share their screen and begin presenting. You have 15 minutes to present and try to leave a little time at the end for questions. And once you're done presenting and it is time for questions, if you could stop sharing your screen, that would be great as well. And so for the audience, if you have any questions, please feel free to ask at the end using the chat function or your microphone. So up first, our first speaker is Shawana Moore from the School of Nursing. Their faculty advisor is Rick Zoucha, and they will be presenting their project, Understanding Perceptions, Beliefs, Values, and Meaning of Mental Health in Young Adolescent African American Females Residing in Low-Income Resource Urban Settings, a mini-focused ethnography study.
Thank you, Sarah. So as Sarah mentioned, my name is Shawana Moore, and I am a Ph.D. student from the School of Nursing. And I had the honor and privilege to work on a qualitative study this year focusing on the cultural perceptions, beliefs, and values and meaning of mental health among young adolescent African American females who reside in low-income or resource settings. And it was a mini-focused ethnography study. So learning outcomes for this presentation are as follows. You'll be able to understand some background information on mental health among young Black women. You'll also be able to explore some of the findings from this mini-focused ethnography study that examined cultural perceptions, beliefs, values, and meaning of mental health among young Black women. So my faculty advisor or mentor for this qualitative study was Dr. Rick Zoucha from the School of Nursing, and my faculty advisor for the Ph.D. program is Dr. Allison Coburg, and she's also from the School of Nursing. So the purpose of my research was to really understand the cultural perceptions, beliefs, and values and meaning of mental health overall within young adolescent African American females who lived in low-income or resource settings, specifically in urban communities. And my research question focused on what are the cultural perceptions, beliefs, and values and meaning of mental health in young adult African American females residing in low-income resource urban settings. So just some background information on why this topic area was important to explore. So we know that based on the literature and data that there are mental health disparities within the U.S., specifically with women. And then when we look at women based on racial or ethnic makeup, we know women of color do have a higher incidence of mental health disparities, specifically within the U.S. And when we look at comparing male and female gender based on studies, we know that females are twice as likely to experience mental health disorders, specifically depression and post-traumatic stress disorder. Also within the Black or African American community, specifically among women, the literature does disclose that there is a high incidence of stigma that's identified, and that really serves as a huge barrier from preventing the specific population of women from seeking mental health services. So the methods that were utilized for this qualitative study, it was a mini-focused ethnography design. There was a demographic survey developed to get characteristics of the overall population that was interviewed. Also, we developed a semi-structured interview guide to help guide the interview process. Also, Leininger's 4 phases for data analysis was utilized to analyze the data after collection. So this is a description of what Leininger's 4 phases of data analysis include. So specifically for this mini-focused ethnography study, we were able to get to the 3rd phase of the study. So phase 1 is analyzing detailed raw data. Phase 2 includes data coding and classification. Phase 3 is identifying recurring patterns. And finally, phase 4 is the major theme. So our hope is that we will continue the study to become a full-scale study so that we are able to determine if there are any more patterns after we can collect more data and hopefully get to the major themes from that data collected. So the results of the study. So participants that were included were age ranges from 22 to 24. The locations in terms of where they resided was in the city of Philadelphia, the state of Virginia, or the state of New York, and they all were college educated. So our initial findings were we had 10 categories based on the analysis of the data, and then from those 10 categories, one pattern was developed. So in terms of the 10 categories, these were the 10 that were noted based on the data. That included avoidance, depression, generational differences, insurance and costs, life transition, pressure, expectations, prioritizing self, stigma, supportive friends, and treatment. So in terms of avoidance, the participants did disclose that the topic typically wasn't discussed within their household setting, and if it was discussed, if the topic was brought forth, they tend to kind of not want to talk about it, so they never were able to move the discussion forward. Depression was one of the categories that 2 out of the 3 participants did mention. Three of the participants did discuss how there was a generational difference in terms of them being willing to want to talk about mental health, wellness, but their parents and grandparents not wanting to talk about it. 2 of the 3 participants did talk about insurance and costs serving as a barrier, preventing them from either obtaining services or having them stop or delay services because insurance or cost of the actual services. The other thing 2 of the 3 participants discussed was experiences with life changes. So a lot came up with transitioning from college to the workforce as a period of when they noted changes in their mood and actually needing support in terms of mental health wellness. 2 of the 3 participants did discuss how they put a lot of pressure on themselves or within their family setting in terms of having expectations for them to exceed well. The other thing was prioritizing self. So 3 of the participants did talk about not always putting themselves first and making sure they were overall well. The other thing was stigma that came out with 3 of the 3 participants discussing areas where they had discussions with either family or friends about mental health or trying to talk about mental health. And there was a lot of stigma around the discussion with mental health. The other thing was supportive friends. So 2 of the 3 participants did discuss having friends that were very supportive with talking about mental health within their friendship. And finally was treatment. So treatment did come up with 3 of the 3 participants as a means or a venue to use to help support their mental health well-being. And from the analysis of the 10 categories, the pattern that came up was pattern of barriers to addressing mental health. So implications to nursing. So this was a mini focus ethnography study, and our hope is to have it to a full-scale study, which will hopefully lend itself to these following implications. Research, practice, and education around mental health, wellness, and well-being within the African American community, specifically looking at young adolescent women. And a conclusion, so again, hopefully we will be able to have this study become a full-scale study so we can go through all the 4 phases of Lineker's data analysis. This will help us identify additional patterns and really identify the meaning of the themes that are describing the phenomena. And I'm happy to answer any questions.
Hey, Shawana, how you doing? Uh, my name's Luke, and I'm in the clinical mental health counseling program here at Duquesne. So thank you for your presentation. Very interested in what you had to say. So my question is, like, in your opinion, what do you feel are some um, changes on a micro level that a counselor could make, and maybe like a more macro level, like societal, that could help address these disparities that you talked about?
Excellent. Thank you for your question and your engagement, Luke. So I think that's a really good question. So I think the biggest thing from like the counselor level, I think is normalizing a discussion with patients, right? About mental health and well-being within discussions, right? Because I can tell you from my personal experience as a Black woman, coming from that community growing up, mental health wasn't something that we talked about or prioritized. And if we did talk about it, it's almost as if it was a taboo topic. Like, we don't talk about mental health, that means something is wrong with somebody. So I think as counselors, it's normalizing a discussion. It's the same. I look at it as if I have a patient who has high blood pressure or diabetes, it's a condition that you treat, right? Or a condition that you try to prevent. So how do we support that? So I think it's making it a normal part of a person's well-being, that mental health should be included. So I think counselors can normalize the discussion within the populations that they serve and encourage individuals to seek services even before something happened, right? So like for me personally, recently, even going through this study, I said, you know what, I should probably have a therapist. So I reached out and got a therapist. I said, we don't have to wait for something to be wrong to get a therapist. It's like going to get your well person exam. You go to make sure everything is well and to prevent illness. So I said, for me, I need to have a therapist. So when something potentially does go wrong, I already have that support and I have those coping mechanisms already in place. And I think on a societal level, I think as healthcare professionals, counselors, community leaders, again, I think having workshops that help normalize the conversation, right? Having different programs within specific systems, whether it be a school system, a community health center, or just a community-based organization where people can come to discuss mental health services or what mental health means. So I think it's like normalizing a discussion on all levels, and just changing the direction on how we view mental health within any population.
Thank you.
Absolutely. You're welcome. I'm happy to take any other questions. Okay, well, I appreciate everybody's time and attention and engagement with my presentation, and I hope that you took away one pearl from it. I can give my email address in the chat if you ever want to follow up with questions as the study continues.
All right. Thank you. Next up, we have Yashika Kamte from the School of Pharmacy. Her faculty advisor is Lauren O'Donnell, and they will be presenting on viral infection in the pediatric brain leads to a loss of mid-stage and differentiating neural stem cells.
Hello everyone, good morning. Today I'm going to talk about how viruses can affect the brain during childhood and specifically what they do to neural stem cells. So, a childhood period is an overall period of development, and that's true even for the brain. So, a number of developmental processes are happening during this time. And if you can imagine, if the viruses have the ability to enter and infect the brain of a child during this period of development, it can not only lead to some of the immediate effects of an infection, such as encephalitis or seizures, but it can also be a reason for development of long-term consequences well into adulthood. Some of these long-term consequences are motor deficits or memory impairment. And this can be because of damage or alteration of some of the developmental processes occurring during childhood. And hence, many of the childhood viral infections in the brain have been seen to cause these long term consequences. And this also depends on the type of virus infecting the brain, the area of the brain getting damaged, and the severity of infection. In general, apart from the brain as a whole developing during childhood, you also have individual cells of the brain that change constantly as we grow older. And one of them is neural stem cells or NSCs. This pool of cells decline as we grow older, and in the adulthood, they are restricted to only specific areas of the brain. During the developmental period in our childhood, the neural stem cells are responsible to build the brain and help maintain the brain architecture. Specifically, these stem cells have two important functions. They undergo proliferation to form more stem cells, and they undergo differentiation to form a stem cell so as to maintain the pool and to give rise to neurons and glial cells. As you can see, these cells essentially build your brain. So it is important to understand what happens to this pool of cells during viral infections, especially during childhood. Now, in general, viruses can affect stem cells in two ways. One is where the virus directly infects the stem cells, and the other is a more indirect way where the virus can infect other cells of the brain, like the neurons. This leads to activation of the resident immune cell of the brain and infiltration of the peripheral immune cells into the brain. The goal of these immune cells is to get rid of the virus and to clear the infection. But the inflammation that it produces during this time can also affect the stem cells. In my project, I'm looking at this indirect effect of the antiviral immune response on the stem cells, but the stem cells are not really infected. To do this, in our lab, we have a measles virus mouse model. Now, mice naturally cannot be infected with measles virus. So, the animals that we have in our lab are genetically engineered in such a way that they express the receptor for measles virus, which is CD46, only on mature neurons. So, neurons are the ones that are getting infected in our model, and stem cells are spared from any infection. And thus, any change in the stem cell pool will now be because of the antiviral immune response and not the infection directly. The images here are also showing the same thing, where in panel C, we can see the neurons in green getting infected with the virus in red. Whereas in panel F, we see that the stem cells in green are spared from infection. Our model allows us to study age-dependent differences of a viral infection in the brain. But in my project, I specifically asked the question as to what was happening to the stem cells after an infection at the pediatric age. To get to this answer, I first wanted to understand how these pediatric mice respond to an infection. Essentially, do they become sick? Do they die? Do they survive? Like, what is happening with them? To do that, I started infecting the pediatric animals with measles virus, and I observed them daily for signs of sickness or illness. I saw that some of the animals were becoming sick, essentially showing some of the neuropathological outcomes seen, like seizures and even problems with movement. And ultimately, they died. But some animals did not become sick at all and survived the infection. This graph here is my survival curve, and on the y-axis, we have the percentage of survival, and on the x-axis, we have days post-infection. The group in white here is a control group, and as expected, these animals do not become sick or die. When infected with a lower dose of the virus, the pediatric animals showed about 22% mortality. When we infect them with a higher dose of the virus, they show about 75% of mortality. And so, to carry out my rest of the studies, we selected the lower dose of the virus to be appropriate because this group gave us a good balance between animals that were becoming sick and died, but also gave us a good number of survivors that we could follow long term to look at some of the long term consequences of childhood infections. In order to carry out the rest of our studies, we did something called as flow cytometry. Now, this technique allows us to count the number of brain cells. So after infecting the animals, I harvested the brain of these mice, and I specifically dissected out two regions. One is the hippocampus, and the stem cells in this region are responsible for memory and cognition. And the other is the subventricular zone, where the stem cells are responsible for sense of smell. Within these regions, I looked at the stem cells, the neurons, and the virus. And in the rest of the brain, we looked at the immune cell profile. Based on my survival studies, in my project, I developed this timeline where at 10 days post-birth, which is the pediatric animals, I started infecting them with the virus, and this is day 0. And I ended my studies at 90 days, which is a long-term time point. In the first 15 days after infection is where I was seeing that the animals that are sick are dying, and the remaining that remain alive after 15 days are our survivors. For my project, I selected 3 different time points to study what was happening in the brain. But for today's talk, I'm going to focus only on the early time point, which is 9 DPI or 9 days post infection. And at this time, I have both animals that are sick as well as animals that are not sick. The very first question we asked was, where is the virus present in the brain and what are the regions that are getting infected? As I spoke earlier, we looked at the hippocampus and the subventricular zone, but we also looked at the olfactory bulb to see if the virus was present. This region of the brain is responsible for maintaining or your sense of smell, and it's actually anatomically connected to the subventricular zone. So the stem cells from the subventricular zone migrate to this region and help in the sense of smell. In all of these 3 graphs, we see that the y-axis is the percentage of total cells, and on the x-axis, we have the control group in yellow and the infected group in red. And we see that the virus is present in all the 3 brain regions. This image here is showing a brain representation of infected pediatric mice with measles stained in red and all other cells stained in blue. The white arrows are indicating regions of infection, and when we zoom in to these areas, we see that the neurons are infected in this fashion. And this told us that pretty much every region in the brain is infected in these animals, irrespective of whether they were getting sick or not. So the next question we asked was, how is the immune response like in these animals at this time? Today I'm talking only about the adaptive immune cells, and your adaptive immune response is generated to specifically fight off the pathogen in hand, in this case measles. We looked at the CD4 T cells, which are helper T cells, and they help orchestrate an immune response. We also looked at the CD8 T cells, which are killer T cells, and they kill off infected cells. And the B cells are antibody producing cells. All of these 3 cells were present in the brain after infection compared to the control group. And this made sense to us because now you have the virus in the brain, so you also need your immune cells to enter the brain and get rid of the virus. So next question we asked was, what is the impact of this antiviral immune response on the stem cells, as the stem cells itself are not infected in our model? To look at stem cells, we used a marker called as nestin. Nestin is a protein expressed by these NSCs. In both of these graphs, we have the y-axis, which is the percentage of cells, and the x-axis is control group in yellow and the infected group in red. Each dot here represents one hippocampi from each animal. So in both the hippocampus and the subventricular zone, we saw a drastic decrease in the stem cell pool after infection. And all this was telling us that the number of the stem cells are decreasing. However, postnatally, the stem cells do not exist as a homogeneous population. In fact, they are present as a heterogeneous pool of cells ranging from being most undifferentiated to stem cells starting to somewhat differentiate and stem cells early in the differentiation process. These cells are much closer to forming other neural cells. So, based on our previous studies, we didn't really know whether the antiviral immune response was detrimental to specific stages of NSCs or was it just decreasing the entire population. In order to get a better handle on this, we started using multiple other markers to try and segregate the different stages of NSCs out. When we looked at the hippocampus, we saw that the most undifferentiated NSCs were not getting affected, but the mid-stage NSCs and the NSCs early in the differentiation pathway were the ones that were getting lost. And this told us that the stem cell pool that was decreasing in the hippocampus was because of the mid-stage NSCs and the NSCs in the early differentiation pathway. The story was similar even in the subventricular zone, where the undifferentiated NSCs were spared, but the mid-stage NSCs and NSCs early in differentiation were getting lost. So the big question that remains now is what is happening to these stem cells? Because we know that the antiviral immune response is detrimental to only specific stages of NSCs and not the population as a whole. So, what I think is happening is after a pediatric CNS viral infection, in our model, we have the neurons that are getting infected. In response to this, the innate and adaptive immune cells are coming in to get rid of the virus. And the inflammation that it produces during this time leads to a loss of mid stage and differentiating NSCs, but the undifferentiated NSCs are spared. Now, one possibility is that the cells that are getting lost are actually dying. But what we saw was, in fact, this was not the case. When we started carrying out long term studies, when these animals are only surviving, that is 3 months post infection, we saw that it is possible that the cells that were getting lost were moving towards the pathway of differentiation and forming different neural cells based on the region we were looking at. I did not have time to show this data today, but what we saw was that in the hippocampus, there was an increase in the oligodendrocyte progenitor cells, or OPCs, and in the subventricular zone, we saw an increase in mature neurons, indicating that it is possible that the cells were getting lost early in both of these two regions, were becoming different cells long term. Now, we know that these two regions have different functions. And what I'm currently trying to understand is whether the alteration of the cell population leads to any changes behaviorally. Some of the things that is commonly seen with human infections, like memory impairment or even motor deficits. With that, I would like to thank my advisor, my lab mates, the animal facility, and our funding. And I would be happy to answer any questions.
I did have a question. So you said that the mice aren't naturally susceptible, correct?
Are not naturally susceptible to measles.
So do you think that infecting them with the virus, the effect is going to be worse because they don't have any like natural immunity or something that might be passed on?
Right. So with measles, at least, and with most other viruses, you don't generally have a natural immunity to specific pathogens to fight off specific pathogens. When we are exposed to a specific virus, that's when our body develops an immunity against it, and we can fight it off better next time. So I don't necessarily think infecting them because they're not naturally susceptible leads to worse off infections. Because if you can imagine a newborn child who's never experienced any kind of viral infection, if they are exposed to any virus, the instinct of the body is going to be, I have not seen this before, so I have to try and fight it off in whatever mechanism I know. And that's similar to what we see even in mice, even though they have not experienced this virus before, which naturally we won't. But when we do experience infections or we are exposed to a pathogen, our body tries to modulate and develop an immune response against it. And that's because of specific viral proteins that every organism does recognize, and we can fight it off. Does that make sense? And does that answer your question?
Yes, thank you. All right. Well, if there are no other questions, thank you so much for that presentation. Next up, we have Odile Enslen from the Forensic Science and Law program. Their advisor is Dr. Lyndsie Ferrara, and they will be presenting their project, Suicide Rates and Trends in Allegheny County during 2010 through 2019.
All right. All right. Can anybody hear me? Oh, perfect. So My camera is not working, so I will not be presenting with a camera. But hopefully I can figure out how to share my screen.
So this is Mary McConnell. To share your screen, there's a little icon in the top right that looks like a square with an arrow pointing to the right.
So I keep on pressing it and nothing's happening.
Okay. Let me see if I can manually do this for you. Okay, apparently not.
If anything, if the person after me wants to go ahead, I can always go after them and figure out what's happening with my camera and stuff like that.
So you said nothing happens when you push the button?
Yeah, nothing happens.
So it might help if you log out and log back in if that's why your camera's not working. It could be browser related. It could be your permissions too.
Okay. Yeah, so I can always take the time to figure technology out, and if the person to just keep everything going, the person after me can go ahead.
Okay, so that would be Kira. Kira, are you ready to present or do you need a few minutes?
I'm ready to present now.
Okay, so we could do that. We could go out of order. Sarah, if you want to introduce Kira for us, that would be awesome.
Yeah, absolutely. Okay, so instead, we're going to hear from Kira Hurley, who is also in the Forensic Science and Law program. Her advisor is Dr. Pamela Marshall, and she is going to be presenting her project, an enhanced DNA extraction method for charred human remains using pressure cycling technology.
All right. Hi. So as she said, my name is Kira Hurley from the Forensic Science and Law program, and I will be presenting on an enhanced DNA extraction method from charred human remains using pressure cycling technology. I was advised in this project by Dr. Lisa Liedvigo, Dr. Angie Ambers, and Dr. Pamela Marshall. So when I started this project, I had two research questions that I wanted to answer. Can PCT increase DNA yield and burn human remains? And can PCT enhance the quality and reduce inhibitors that may be present in burn human remains? So for these questions, I had some predictions. Initially, I was thinking that PCT would increase DNA yield and burn human remains in order to answer my first question. And for my second question, I predicted that PCT would enhance the DNA quality while simultaneously reducing the effect of inhibitors on these challenged samples. So I've mentioned pressure cycling technology, or PCT. So what exactly is it? So pressure cycling technology uses a barocycler, which uses alternate high and ambient hydrostatic pressure. And this can be used to alter the conformation of inhibitors that may be present in a sample. So the pressure actually enhances the hydrogen bonds that are present in the DNA structure, while altering the conformation of the bonds that may be present in PCR inhibitors, such as metal ions like calcium that is present in bone and teeth. So a little bit about the relevance of this project is for the identification of bodies compromised by heat. So when you have a mass disaster event like a wildfire or a terrorist action or something a little bit smaller scale like a specific arson, the remains that are from those results can often be extremely degraded to the point where only bone and teeth remain, making identification sometimes nearly impossible. And we really saw this with 9/11 terrorist attacks. There are still over 1,000 remain segments that remain unidentified. These samples are all severely degraded, and there's widespread contamination in all of the samples due to all of the building materials that were present at the scene that day. So all these inhibitors, the ones that are present in the matrix itself, so for teeth and bone, the calcium ions pose a large issue. With teeth, you also have the additional issue of metal fillings, which can also pose downstream PCR inhibition. So looking at this, I wanted to try and develop a method that can maybe help remove some of these inhibitors to produce a higher quality profile for these remains for identification purposes. So when I started this project, I had human metacarpals that I was cutting into 3 segments using a Mopec autopsy saw. And then additionally, I had later another sample type, which was human teeth. These were stored in 100% ethanol. I was able to develop 27 profiles for my teeth, deciduous and adult teeth. I lost 3 samples along the way due to just vials cracking. So what I did is I took my teeth and I burned them at one of 3 temperatures, 100, 300, or 500 degrees Celsius for 30 minutes. Part of the reason I chose these temperatures is because your average bonfire is about 400 degrees Celsius, your average house fire can be around 600 to 700 degrees Celsius, and then your average car fire can be around 900 degrees Celsius, depending on different accelerants are present. So I wanted to try and look at that lower range to start, and then possibly expand on it later. After all of my samples were burnt, I then powdered them using a freezer mill. Once all the samples were powdered, they then underwent an extraction process. I utilized the University of North Texas Center for Human Identification's demineralization extraction of skeletal remains protocol. And this protocol is a PCI extraction with an overnight incubation period to help demineralize any of the sample that may be present. So the calcium ions that are present in bone and teeth, just to try and get that out of the sample as early as possible. So for half of my samples, there was an additional PCT step that was underwent on the barocycler by Pressure Sciences Technology. So what this was 30 cycles of alternating pressure, which was 20 seconds at 35 kpsi and 10 seconds at ambient psi. And additionally, I had reference buccal swabs that I processed using the Kinamp DNA mini kit in order to compare the profiles developed from my teeth samples to see for allele dropout. So for my analysis, I had a quantification step, which I used the Quantifiler HP kit on a Quant Studio 5. After my samples were quantified, I then normalized them, and they went amplification using the Globofiler kit on a GNAMP 9700 thermorecycler. After the samples were amplified, they underwent genetic analysis on an Applied Biosystems Seq Studio genetic analyzer, and then I interpreted those results using SoftGenetics GeneMarker HID software. So initially I had bones that I was using, and the bone results were not what I was looking for initially. So the bone samples yielded far less DNA compared to the teeth samples, as you can see here, both at, so especially at the small autosomal DNA quantities, I had 0 from the bone, and then for the tooth, there was actually 0.21 for the first initial tooth that I processed. Part of the reason that we think that the bones didn't yield any results is due to a possible PCR inhibitor due to a chemical preservation agent that may have been used on the bones before they were taken into our custody. Because of this large difference in DNA, we actually switched our matrix type from bone to teeth. So from here on out, I will be talking about teeth samples. So at 100 degrees Celsius, the PCT enhanced method yielded more DNA on average compared to the normal extraction method. This was a significant difference at the large autosomal DNA section, whereas at the small autosomal DNA section, the results could not be considered significant after a student's t-test. Then you can also see at 300 degrees Celsius that the large autosomal DNA, the normal extraction yielded more, but this was not a significant difference. And then at the small autosomal DNA, the PCT enhanced method yielded more, but again, this was not a significant difference between the 2 methods, even though if it was yielding higher amounts on average. At 500 degrees Celsius, we saw the total dropout of large autosomal DNA in both the normal and the PCT enhanced extraction method, signifying that we probably weren't going to get any results from the samples burnt at these temperatures downstream. So for the small autosomal DNA, though, the PCT-enhanced method did yield more, but it was not a significant difference compared to the normal extraction method. So additionally, with my quantitation results, I was able to calculate the degradation index, which is the amount of small autosomal DNA divided by the large autosomal DNA. I wasn't able to calculate this for all of my samples, as the large autosomal DNA frequently was unable to be determined in many of the samples, reducing the sample size for this graph. So as you can see, it was showing that the degradation index was decreasing as temperature increased, which was opposite of what we expected to find, as the increased degradation index means that the sample is more degraded. Part of the reason that I think we were seeing this negative slope was because so many of the samples were undetermined for the degradation index. And also you can see that there is a very low correlation for both of the normal and the PCT-enhanced extraction method in this calculation. So moving on to the electropherograms. On the top here I have the tooth sample, and on the bottom you're going to see the reference profile. So for my sample extracted at 100 degrees Celsius using the normal method, there was a 5% dropout rate of alleles at the TPOX allele, and you can see the starts of a ski slope effect showing that as the alleles got longer in length, the larger loci, the intensity of fluorescence for the profile started to decrease. At the same temperature, using the PCT-enhanced method, we had all 40 alleles that were present in the reference sample, so a 0% dropout rate. And we additionally had one unknown allele that dropped in this sample. Important to note is that we believe this allele drop-in is due to elevated stutter products, as the the drop-in allele fell in a stutter position, and it was not in any of the other reference or exclusionary samples taken. At 300 degrees Celsius for the normal extraction, we had a 100% dropout rate of alleles. At 300 degrees Celsius for the PCT-enhanced method, we had 6 alleles show up, so that's an 85% dropout rate compared to the reference. There were 7 unknown alleles that dropped in. 5 of these alleles fell in elevated stutter positions for either the 1 or 2 stutter. At 500 degrees Celsius, we had the total dropout of alleles, which was expected, as we saw that there was no large autosomal DNA at 500 degrees Celsius, and this also held true for the PCT-enhanced method. So some of the conclusions that I was able to see is that PCT-enhanced methods can assist in yielding higher quantities of DNA on average compared to the normal extraction method that was used. The difference is not significant in the quantity except for the large autosomal at 100 degrees Celsius. So the PCT-enhanced methods can also yield higher quality profiles, and this was indicated by the fact that we were seeing less dropout in the PCT-enhanced method compared to the normal extraction method in the profiles developed through the allele dropout rate. We also saw that as temperature increased, there was a lower yield of DNA, and this was to be expected, as many studies have stated that there is a negative correlation between increasing temperature and the amount of DNA that is able to be recovered. So we saw also that at the higher temperatures, there was an increased degradation in these teeth samples, which again is supported by literature from past studies. So we were able to see that profiles could be developed at 100 degrees Celsius. We had a partial profile with the PCT-enhanced method at 300 degrees Celsius, and we saw that profiles were not able to be reliably developed in teeth grown at 500 degrees Celsius for either the PCT or the enhanced extraction method. I'd like to acknowledge the Duquesne University Forensic Science and Law Program, Sydney Reed, Dylan Baxter, Dr. Lyndsie Ferrara, Dr. Jennifer Hammers. And if you have any questions, let me know.
All right. If there are no questions, thanks, Kira. Okay, I don't... Odile, did you figure everything out?
Yes. So I have 2 amazing roommates, and they've been helping me with technical difficulties. So I believe this should work now.
All right. So, once again, Odile Enslen from the Forensic Science and Law program, and faculty advisor Dr. Lyndsie Ferrara on suicide rates and trends in Allegheny County during 2010 through 2019.
Can you see the slideshow?
Yes.
Okay, thank you. Sorry guys for technical difficulties. I guess this forum just hates Macs, so I apologize. But so like Sarah said, my name is Odile Enslen. I am a 5th year within the Forensic Science and Law program here at Duquesne. And my research is looking at suicide rates and trends in Allegheny County during 2010 through 2019. So just a forewarning and a heads up that I will be talking about suicide and suicide methods in depth. So just wanted to give everyone a fair warning. But the reason why I started looking at suicide rates and trends within the impoverished community is because when we're looking at services or even in the literature, there's this gap when trying to connect the two. So within resources, obviously for people who are living in poverty, we have food stamps, Medicaid, EITC, all sorts of things to help people out. And then on the suicide aspect of things, we have safe planning, crisis hotlines, lifelines, therapy, and nonprofits that do amazing work here. But there's none that bridge the gap in between the two. And this is also apparent within the literature, just because when we try to look at suicide rates in the impoverished community, there's no concrete answer on how they're correlated to each other. So this came up with the my research question, which is, are there higher suicide rates within impoverished communities? Which then also got me thinking, are there different suicide method trends within impoverished communities compared to non-impoverished communities? And how do these rates and trends compare to national averages? So my hypothesis was that there is a positive correlation between suicide rates and poverty, and that there would also be a difference when looking at suicide method trends. So because I'm in the forensics program, there has to be a forensic relevance to this. And what better statement to take than one directly from our medical examiner's office here in Allegheny County. So the office said, as I quote, the office has the additional mission of using the knowledge obtained in this process to promote education and research into the key public health problems facing the citizens of the county, such as gun violence, drug overdose, suicide, and other issues. So not only are we trying to promote better resources for our community, but also educate the citizens within it. So the two data sets that I used, one, the suicide data came from the medical examiner's office, and this data was all the suicides that occurred throughout 2010 through 2019. And then my poverty data came from the Census Bureau. So the data that the Census Bureau collects is, there's a lot that they collect. So we all know about the 10-year census that happens every 10 years. But they also do multiple surveys throughout the year. So one of these surveys is called the American Community Survey. And this survey is done every 1, 3, and 5 years. So depending on the location that you're looking at, so for us, we were looking at municipalities, which is a very small population. The 5-year estimate was the best choice for us. So what this 5-year estimate survey does is it looks at the income of households that they survey within the municipality. So what they do is they calculate that household's income by using earnings, rents, interest. So all these things that are listed here in Figure 1 go into calculating the household's income. And that income threshold is then compared to the poverty threshold that the Census Bureau calculates every year. So for myself, being one person under the age of 65, in 2020, if I made less than $13,465, I'd be considered living in poverty. And then the other way around. If I made more, I would be considered not living in poverty. And so that's how the Census Bureau comes up with a rough estimate of how many people within a population are living within poverty or out of poverty. So when looking at Allegheny County as a whole, we see that it's a very diverse location. So what I mean by this is Allegheny County has 130 municipalities in total. And within those 130 municipalities, the poverty percentage ranges from 0.3% all the way up to 60% of that municipality's population living in poverty. And at the same time, we see that suicide cases are on the rise in Allegheny County. In total, there were 1,914 suicide cases altogether from 2010 through 2019. That number did decrease to 1,555 just because we wanted to look at cases that solely came from living in Allegheny County. The medical examiner's office does look at cases that come outside of the county and even the state. So going to answer my first question, if there is a correlation between suicide rates and poverty percentages, this is my correlation graph. So as you can see, there is a nice clustering of the cases, but there's no real strong positive correlation. My correlation coefficient came out to be 0.132. So even though it is positive, it's very weak correlation between suicide rates and poverty percentages. Going on to answering my second question, this is just a breakdown of the causes of death that were used in the suicide cases. So as you can see, firearms and hanging were the top two, followed by drugs as the third. So what I was able to do was I made poverty brackets, so low, medium, and high. Low poverty would be less than 10% of the population was living in poverty. Medium was 10% to 29%, and high was 30% or higher. So what I did is I broke down all of the poverty brackets by the 11 causes of death. And visually, from the pie charts that I made, there visually is no difference between the poverty brackets. So as you can see here, the low poverty bracket, medium, and high, there might be a slight difference by a couple percentages, but nothing that really popped out when looking at them. So what I did is I ended up doing a chi-square test just to see if cause of death and poverty were independent of each other or not. And what came back is that they're actually dependent of each other. So what I did is I took a further look and looked at a table with all the causes of death with the total cases broken up by poverty bracket and then percentages next to. And what I did here is I highlighted a few that just kind of pop out. So when looking at, let's say, blunt force trauma, the low poverty bracket has a percentage of 2.81, while the high poverty bracket has a percentage of 8.70. That, to me, that's a big difference. So that kind of pointed out the dependent relationship there, as well as drowning. Again, in the low poverty bracket, 0.7, medium, 2.2. But then in the high poverty bracket, we saw a 4.35%. So again, they're almost twice as likely to have been used in the high poverty than compared to the medium and low. And there are a couple others highlighted as well. But there obviously were some other key results in this research, but due to time constraints, though, I wanted to point out the ones that really highlighted the results that we got in answering my two research questions. So we did see that though there was a positive correlation between suicide rates and poverty, it wasn't as strong as we initially thought, which we can still learn information from this in helping the community with resources, as well as seeing that cause of death and poverty are dependent of each other, so we can better tailor those resources to the preventing those specific cause of deaths. So I just want to take a few seconds to personally thank everyone that is listed on this slide. they really helped me throughout these couple of years doing this project. It has been a long, long couple of years, but I'm happy that it's come to the end. But thank you all so much for taking the time to listen, and I'll take any questions.
This is Luke. Not as much a question, but a comment. Like you said, although there wasn't a strong correlation, it is still a problem. So I just appreciate you bringing some awareness to it. It's going to be a difficult thing to talk about, but it's important that we do so. So I appreciate your work. Thank you.
Yeah, thank you, Luke, for that comment. That means a lot to me.
All right. Thanks, Odile. And that concludes the presenters for this session. Thank you for everyone who presented and everyone who listened and participated in these presentations.